Hi, everyone. Welcome to Terpenes and Testing, the podcast. This broadcast is brought to you by Mace Media Group and Terpenes and Testing Magazine, the industry's first publication dedicated to cannabis science. I'm Jason Lupoi, and I'm the editor-in-chief of Terpenes and Testing Magazine, and I'll be your host as we navigate across the landscape of cannabis and psychedelics science. The cultivation of cannabis is a much-needed opportunity for several reasons. Obviously, we use the plants to get the sought-after therapeutic secondary metabolites. The scent of fresh cannabis invokes a therapy of its own, and even tending to the plants, watching them grow and nurturing their needs is medicinal. We use cannabis medicinally or preventatively for our health, right? So it's equally important that our plants are healthy. It may seem rather sci-fi, but plants share several qualities with us. Just as plants talk with each other via terpenes, they can also get sick. The trick is to find propagation methods that do not contaminate your genetics over time, weakening your mothers and ultimately taxing your entire operation. So for this episode, I wanted to speak with Dr. Hope Jones. Dr. Jones is Chief Executive Officer of Emergent Crop Sciences, or ECS. She is a graduate of the University of Arizona Controlled Environment Agriculture Center, where she worked to propagate endangered native species using tissue culture techniques. As a NASA scientist, Dr. Jones developed technologies and protocols to grow crops for deep space missions. And Dr. Jones her work currently focuses on optimizing micropropagation protocols and facilities for the cannabis industry. Dr. Jones, welcome to the podcast.
It's really a pleasure to be here. Thanks for having me.
So your bio really intrigued me. I really did not know that you were a NASA scientist, and I was curious if perhaps I'm sure a lot of other folks are wondering what would could you discuss what work you did for NASA and why growing crops for deep space missions was of interest?
My pleasure, because it was by far one of the best experiences I've ever had. Both scientifically but professionally. I loved doing what I did out there in Florida. I was working at the Kennedy Space Center, and our focus there is it was more of a form of what's known as astroculture. So that's a technique used to maintain closed environment systems for plant growth in for example, at the International Space Station, and even just on deep space missions. And what I did was I was newly graduated, and I wanted to grow plants in space. For some reason, this was a good idea for me. And I was doing basic tissue culture at the university, and I just reached out to researchers at NASA that I had a lot of respect for their papers. I talked with my, the professors and PIs at the university, and they said, contact so-and-so. And I just wrote them and I said, this is something I want to do. And they had me out. I gave a presentation, and then off I go. And when I started, it was starting with growing plants that were high in antioxidants. And the reason why they wanted to focus on that, why that was my directive, was because when astronauts are in space, they're exposed to high rates of cosmic radiation. So if they were to consume foods, crops, strawberries was what I was focused on initially, perhaps it might actually lead to evidence that it might increase their internal shielding, right? So just by using fruits that were high in antioxidants, you could think of foods that are high in colors. Blueberries was one of the goals, but it's so woody, it was really one of the top priorities. But there's a lot of great foods, vegetables and fruits that are high in antioxidants, and to develop those systems required plant tissue culture, because everything that goes up into space, every pound costs a lot of money. And so we also wanted to have this as a contained system, a very tightly contained, controlled environmental system, because we don't want anything getting out in the in the shuttles at the time, and then or into the space stations. And we wanted to be able to measure what type of lighting was necessary, what type of nutrients, what type of substrates. We wanted to measure the the volatile organic compounds that were emitted from anything that was to go up with that type of mission. And that was what I was doing. It was amazing opportunity. And I decided that's what I wanted to do for the rest of my life. So if I was going to stay at NASA and lead research teams, I needed to get my PhD. So I went back to get my PhD. But unfortunately, during that time, that was really when there was the brain drain, and funding was no more for life sciences at NASA. And so I still have the dream, though. I still want to be able to grow plants in space, and hell, why not cannabis?
That sounds great. That's really a neat story. And I suspected it was something for astronaut health, but that's really neat, and that's a good point that you make. That it costs a lot of money to take things into space, and so trying to do a tissue culture. So would this have been done on a space station, or actually during the mission? Not something they would have brought with them? Would they have been actively growing the crops on the missions?
That's exactly right.
Okay.
They had prior to my joining the team, they had what the acronym was PESTO, and I can't remember today what that actually stands for, but it was actually to study plant growth and a wheat. I think it was wheat, small architecture wheat up in space. And then this whole idea that antioxidants in strawberries was to be one of them as well.
So I'm curious how that work ultimately brought you to the cannabis industry.
That's a weird journey, but let's just be short about it. I went back, got my PhD in molecular cellular biology with an emphasis on genetics, basically cancer biology, studying chromosome instability. I didn't want to stay in academia. It was not, it really wasn't for me. I didn't want to write research grants, and a PhD is not, today is not what it was, even 20 years ago, where, you would automatically have jobs and what have you. And I really liked the idea of going into industry, but I wasn't necessarily sure what I wanted to do. And there was a period of time where I was doing several different things. But when I was in Tucson, I was working at Pima Community College at the nursing department, running a simulation lab. And I saw an ad in Craigslist, for a cultivation position. And I was like, why not? I was the happiest I ever was when I was growing plants and when I was doing controlled environment ag. And so I didn't get that job. I actually got hired as a CSO and to start a tissue culture program. So that was when I started working with cannabis and tissue culture. So that was probably about 7 years ago now. And boy, that was an amazing experience, not necessarily for the good reasons. There's a lot in cannabis that still needs to be worked out to be true commercial producers. So when I left that position, I decided to start ECS and just do what I could to help the industry.
I'm curious, in my intro, I talked about plants getting sick, and I think in a world still reeling from COVID-19, we may forget that plants can get sick. And I was curious if you could talk about specifically how plants can get sick.
Plants they're sessile. They can't move, right? And so they have chemical defenses, and in order to use that, it requires energy. And when a plant gets either attacked mechanically by pests, and pests are very interested in the sap, they like to get into the phloem. A lot of these pests, so white flies and thrips and mites, because they really like the sugars. They're going after the carbohydrates. So, bugs carry disease. And so that is the main route oftentimes for initial in the wilds infection. And you can also think of even something like a huge dust storm is like hitting plants with sandpaper. and then they have these open wounds, and now you have, you know, just ubiquitous spores around the environment that land on them, and then, you know, that's a really good way to get a fungal or a bacterial infection. And so since they can't get up and leave, they have to divert their, you know, their metabolites, their energy, their resources, because there's no free lunch, to be able to deal with this stress response. So that's one way in nature it happens, but really in commercial cultivation, it comes from us. So it does definitely come from vectors like pests, as far as an introduction of a disease, but mostly it comes from cultivation practices, just by literally moving through the grow. If there's not a, if you're starting in the dirty section, then you go into a mom room, you know, that's not a good cultivation practice to take. And unfortunately for cannabis, the cannabis industry, just due to the clandestine nature of how the industry had to survive for so many years, and why I have a job, thank you everybody, is nobody's going to school to, or nobody's going, you know, to work in these large professional nurseries to grow cannabis. And so really what we're doing is we have been spreading and sharing. This is a industry for since really you could buy it, but you also share with everybody when you have something really great. Really spreading diseases by, you know, here, take this clone, and if this clone's infected, and you put it amongst the rest of your stock in your mother room, and they happen to be overlapping, or you're using shears and you're not cleaning them in between from plant to plant. Well, now what was just a single problem with one plant that, you know, somebody might have shared with you, or that you might have bought, now can actually become quite costly, and really have a huge impact on your entire business downstream from that point on.
Are there specific diseases that are unique to cannabis, or ones that are more common to cannabis than what you would see in other plants?
You know, there's one big one. So there's what I call the usual suspects, and you know, fusarium, pythium, botrytis. Plants have different type of diseases that they deal with than we do. And so there's the usual suspects that a lot of plants are susceptible to, and cannabis is definitely one of them. We also have something in the industry that I think is our own epidemic, and that's hop latent viroid. And so
Right.
viruses of viroids, and once a plant becomes infected, there's no vaccine, there's no cure. It eventually, it will kill the plants, and they can be carriers for a long time before you might even see symptoms. But once that plant gets into stress is when you start to see those symptoms. And, you know, I have to tell you, Jason, this is a plant that has been bred really for two key things: high THC and short, fast yielding architecture. And so this plant has lost what it had evolved to have, a lot of resistance for pest and disease resistance, because again, that's part of all plant evolution. That's why they can survive in the wild and be able to deal with attacks. But unfortunately, cannabis is not that anymore, because of the, you know, the underground cultivation and breeding that's happened over, you know, decades and decades. So now I think of it like we have this plant that is basically an ICU patient, and we release it into the wild, and we're like, good luck. And somehow we're surprised we see it back at the hospital, right? And because it's got no protection. And also the other problem that we have is, you know, the genetics get old, and when plants get old, they stop making certain proteins. Just like as we age, there's a half-life to protein production. So that's why we start to get, you know, wrinkles and sagging skin and things like that. But also, as a plant is exposed to a lifetime of stress and weathering and all of these other issues that happens just by being alive, well, they decline, and that happens because of epigenetic signals, for example, within it. And so it's like, oh, I'm under attack, I'm underwatered, oh, I'm not being fed correctly. And instead of being focused on just growth and development and doing that every time, it's now splitting and sharing its resources, and now it's a tug of war basically. And so you can imagine somebody or a plant, I'm totally thinking of myself clearly here right now, old and stressed. No, but you can imagine a plant that's enduring that throughout its lifetime. When it gets an infection like a virus or a viroid infection, that's definitely going to have a greater impact than if the plant was infected when it was really vigorous and healthy. And so that's been my focus in the industry. I didn't know it was going to be my focus, but it's definitely been the direction of my focus.
Could you talk about what tissue culture means for those unaware, and also how it can help how it helps keep plants healthy?
Yeah, plants are awesome. So in the regards of, well, they just are, but they have we call it totipotency. So that's the ability for a plant cell, normally any plant cell, to regenerate new tissues and organs, and then become a whole new plant that includes, the shoot and the roots. And so with tissue culture, what we do is we actually use that to our advantage. And we say, okay, if we can take small parts, these are really small parts, and we anywhere from, a few millimeters up to, maybe if we're doing nodal production, it might be about an inch, but it's really small. And it's basically the technique of using the cells, tissues, or leaves, or any other organ. And we're doing this in a sterile environment. And then we place those tissues or cells in a sterile vessel that has nutrients. And that nutrient contains everything a plant needs for growth and development. It's vitamins, all of the elements. We provide plant hormones. We're basically steering the plant with what's in that media. We seal it so it's sterile, and then totipotency takes over. So what might just be a cluster of cells, if with the right media formulation, steers into new shoots. And then when you take that same plant that only has shoots and put it onto media that actually stimulates root formation, that's how we can actually generate an identical clone when done right, in a very, very small manner under sterile conditions. And then we can generate and multiply large numbers of that in the same way. So it's not only a way to generate a plant from a few cells or organs or tissues, but then we can actually mass multiply. And tissue culture is basically what we call micro propagation, right? So it's propagation, but using very, very small tissues, cells, and organs.
How does it differ from, perhaps, so-called more traditional ways of propagation like cloning, or, I guess in terms of health of the plant, quality of the genetics, things of that nature?
So with traditional cloning, and traditional cloning will never go away. This is a standard practice in all horticulture and ag. It is for us, and it always will be. But the advantage of tissue culture in doing this is, with conventional cloning, you take one cut, you put it into a hormone, and stick that into a substrate and let those roots form. And then in the end, you get one plant. Here with tissue culture, as I've already said, we can generate from very, very small pieces of tissue a new plant that's a clone. It's genetically identical from the cut of the plant that we took. But additionally, we can then go through multiple cycles of generating shoots. So we can generate one shoot, and its multiplication rate starts to branch. So then we dissect that after a couple of weeks. Those new pieces of the dissection are stuck into fresh media. So one plant is now becoming four. In a couple more weeks, we develop more shoots. And then we do the same process over and over again. So whereas one cut in conventional gives you one plant, here one cut can give us really thousands and thousands of plants. But more importantly, when we do conventional cloning, we're not doing anything about the health and vigor and disease status of the plant. We're just propagating up what we get in that cut. So if we take a cut from a mom that's been infected, Well, damn right that clone is also going to end up infected. So what I do with tissue culture, and why really tissue culture has a place in agriculture in general, above and beyond than just the multiplication rate, is now we can actually start to work to eradicate the diseases that have been infecting that plant. And so that's the first step in my mind of the process. Is we take this in. We start with the meristematic cells, which is think of every node on a plant. Even think of a carnation, like where the leaves are. When you peel those leaves off, that's a node of that carnation, right? In cannabis, we have tons of nodes. There's a lot of branching. And at every tip on the roots, we have nodes at every tip. So we can harvest that. It's very small. We use the dissecting microscope. And the reason why we do that is because most of the fungal and bacterial and a lot of the viral diseases require the vascular system as their mode of transport throughout the plant. And that's how it becomes a systemic problem. So the meristem is actually very cool because just like our stem cells, generally speaking, well, the stem cells are not defined as anything. They're undefined cells, same with plants. So the meristematic cells are undefined, not attached to any particular vascular part yet. And so if we carefully dissect that off, place that on the media, well, then now we have cells that were not at all infected with any of the disease particles of the rest of the plant. And then we go on with the process of steering and getting shoots and roots and generating healthy plants. But sometimes it's not that easy. So that's the advantage and the difference between cloning and TC. So maybe I'll just stop there because then I'll get into rambling about other things. But that's the advantage over cloning. Now, once you have clean stock, from tissue cultured plants. So let's say a grower wants to go ahead and get some new disease, at least what's testing negative for any of the diseases that a tissue culture lab was testing for, and generate a mother from that. Then when you take cuts off of that mother for your vegetative production and put it into your prop cycle and into your grow, well, now you have really healthy, vigorous plants. And the main difference, the cost it is for cultivation at that point is going to actually reduce, because you don't have the same costs that it takes to grow a healthy plant as the cost is to grow old diseased plants. You also have uniform cultivation. Everything is predictable, and then you should actually start to see real commercial like propagation and then yields, because everything is behaving as it should. You should have, consistent and uniform chemical profiles, time to yield. And as a matter of fact, oftentimes a healthier plant requires even less time in veg. So there's a lot of advantages for doing it. And, those are just a few.
Do you think that tissue culture has kind of started to sweep across different grows across the country, or do you think that a lot of grows still haven't switched to a method like this and are still, kind of doing things in a traditional way and maybe kind of suffering the consequences from that?
I think all of the above, actually. And when I started giving, I can remember when I gave my first presentation at one of the conferences, several years ago. Nobody even believed when I was talking in the audience that plants could get infected with virus. They didn't think that. So as the industry expands and learns, and as companies become, more and more informed, then they are looking for solutions, and tissue culture is one solution. I think another solution is we need better genetics, and we need to be breeding. But why everybody hasn't done it is it's a serious venture, and there's not a lot of companies there's not a lot of companies like mine. And so the resources are very, very limited for companies to purchase clean stock from companies. So, and then there's companies that do want to do it in-house, and I consult and help them get going. But one of the challenges has been is a lot of the companies don't understand the ramp up it takes to do tissue cultures. So everything that I've explained about doing TC and, the time doing the meristem and doing this, there's a ramp up time. And that's a real problem for a lot of companies because they want an immediate ROI. They want immediate gratification. This is a business, right? The margins are getting tighter. And so oftentimes companies will kind of half-ass it. Or the other issue is the tissue culture companies out there. Well, we're all self-funded, right? There's no academic research behind ours, not yet anyway. And it's going to take a long time before that research is there and caught up. So I've spent the last 5-6 years with an R&D lab. I may have started cannabis tissue culture a long time ago, but just because we can culture it doesn't mean that we're doing it all correctly, optimally, or even at our own proper economic scale, right? To be able to monetize it, but also the testing that's required. So when we do tissue culture, wouldn't it be nice to do a test to see if in fact we did get rid of the pathogen or the virus? Well, that costs money. So there's a lot of reasons for the delay for tissue culture, a lot of good reasons, valid reasons, but it's absolutely one of the solutions for the industry, and more people are looking to it.
Yeah, I think what you said about that fast return on investment is something I've seen across most aspects of the industry, where a lot of times people are not thinking downstream 5 years, 10 years, and they're looking at sticker price up front of a given piece of equipment or a technology. And it sounds like from our conversation here that there's scale is definitely there, because you said a single cut could potentially produce 1,000 plants. A cost perspective, you're cutting costs at the same time, but there might be that initial investment to kind of get things off the ground and dial it into a level that's acceptable to have healthy plants for longevity. So that's something I've definitely seen across a lot of aspects of the industry as well.
Yeah, absolutely. And I will tell you myself personally, one of the things that I'm going to take on like a maniac is the idea of, to do a tissue culture lab, at least in Arizona, we need to be under a cultivation license. That's precious square footage. I mean, really the whole point of tissue culture and agriculture is for those producers to devote every aspect of their square footage to something that's going to contribute to their bottom line. Propagation does not, mother stock does not, tissue culture labs do not. Processors, kitchens, right? All of those things do. So that's a real challenge for tissue culture companies right now, is how do we do this? Because again, being self-funded and in Arizona to get a license, I mean, boy, minimum 10, 15 million. This is not a real possibility. So that requires partnerships. And then we get into a whole different hot, hairy mess, don't we?
Yeah. Well, we're almost out of time, and in a world filled with misinformation, I always like to kind of myth bust, or do our share to myth bust things about the cannabis industry. And I was curious if you could share one main misconception that you see in here regarding cannabis cultivation.
I've been thinking a lot about cannabis and cannabis sustainability. I do think that it's a misconception that cannabis is going to be better than other crops like cotton, for example. And unfortunately, that's just not true. And I think we need a lot of research to go into that. However, the other thing that isn't talked about or is just completely skipped over when it comes to climate change is the idea of how cannabis actually really is an amazing sequester of CO2 and binding that carbon within itself. And I really do think cannabis could be an amazing solution if we actually took the time to look into it. And so, yeah, good question. I mean, I was just talking with somebody about that earlier today, and I was like, I wish we actually were able to do more of that research. And so R&D, baby, that's what we need to do to get rid of any of the misconceptions when it comes to cannabis. And I think that will also help on a policy level.
Yeah, no doubt. Well, Dr. Jones, I want to thank you for taking some time to talk with us about the benefits of tissue culture today. It really was a great conversation.
Oh, it's my pleasure. Again, thanks for having me. And you have a good one now.
Thanks to all of you listening for tuning in. We will see you next time on Terpenes and Testing the podcast. Until then, please come visit us at www.terpenesandtesting.com or on one of our social media platforms. Cheers.